that this was actually at the end of his lab. So his lab here existed, and this is the lab website. You can see it says unearthed and not, it used to say unearthing secrets of the microbial world. Now it's in past tense because the lab actually closed. I was actually one of the last scientists left standing in the lab. But from 1983 till 2018, for over 35 years, the lab was open, and Roberto trained, I think, around 150 different microbiologists that went off to work around the world. So what was partially so special about this lab, and it was a dream lab for me to work in after my PhD, is that it was very wide and focused. So maybe you can even tell from this screenshot of the website, Secrets of the Microbial World. It's a very broad topic, and he brought in a lot of eclectic people and eclectic projects to work on in the lab, all under the general theme of microbiology, but he had wide-ranging interests, and that really made it kind of a special lab where there were many people working together, but also working on diverse projects. So I already mentioned a few times that I'm a microbiologist and showing you a little bit about the lab that I came from. But I want to also talk about what the microbial world is. So hold on one second. Oh. This is actually, sorry about this. I don't know what happened. I lost my presentation. Stand by for 1 second, sorry. No, I'm actually going to close this. Okay. So, yeah, let me see. So actually what happened is I've got a different version of this PowerPoint that is not the right one. And I really apologize, but I'm gonna have to pause for 1 second and just fix this because I can see it's a pretty big difference. So sorry about this. Just give me 1 minute and I'll promise to make up for it. And Okay, I'm back and I fixed it. And I apologize again for that. Let me make sure you can see my screen again.
We can see your presentation again.
Okay, so sorry about that. I don't know, the Dropbox file reverted to a previous version of the talk, and I could see enough that it was going to be different enough that I couldn't swing it. So anyway, so I'll go a little bit more quickly now through just this background. So I was in the culture lab studying microbes. So for anybody who doesn't know what microbes are, so I don't want to just take for granted that everybody knows about microorganisms. So when we say the microbial world, or like when Roberto has on his website, Unearthing Secrets of the Microbial World, this is the microbial world to me. So this is a video that I took of some water from moss. So this is a little bit of the film of water that's on top of moss as you're walking around. It could be any piece of moss, and you can see all these little microscopic creatures. This right here is called a rotifer. It's a type of microscopic animal. There's some water fleas, and then even smaller than that are bacteria and other microbes. So this is what you should sort of think of when you hear this kind of poetic term, the microbial world. It turns out that most of the biodiversity on Earth is actually bacteria and other microbial species like that. So these are all species that are too small to see with the naked eye. So that's really what unites them. So in the most basic definition of what a microbial species is, or what a microbe is, we're talking about any organism that's too small to be seen with the naked eye. So these here are some bacteria that are living in a community called a biofilm, and they're all attached to each other through this sticky substance here that's called the extracellular matrix. So this is really what this microbial world is, really what contains most of the biodiversity on the planet. And I'm not going to talk about how we know that, but I'll just say it's mostly based on genetic information. When we survey natural environments and look at all the DNA that's present there, all the genomes that are present there, we can see that there's this tremendous diversity of microorganisms in just about every site across the planet. So also in this quick intro to microbes, microbes have existed since the beginning of life itself. Actually, it's maybe better to say that microbes are the beginning of life itself, because the first organisms on the planet that were first to arise on Earth were microbes. But they were just discovered by humans a couple hundred years ago. So I always like to introduce that, because I think it's really neat that microbes have always been here. They've been on the planet for as long as we've been human, but they were just discovered in the late 1600s. They were discovered by this person here. His name is Antony van Leeuwenhoek. You might have heard of him already. He's pretty well known as one of the founders of microbiology. And he sort of accidentally discovered microbes because he had been making these little metal simple microscopes. The part of the microscope that magnifies is this little spherical lens right here. And he was doing this because he was a textile merchant, and he was aiming to look at the quality of the fibers and the textiles he was purchasing. But he actually accidentally discovered this whole microbial world. So if he took a little bit of pond water or moss water, like I showed you before, put it on this little pin right here at the top in front of the lens, and that's how he discovered all these microbes. So he was the first person to discover bacteria, red blood cells, microscopic animals, all sorts of microorganisms. Because this was a long time ago, this was before cameras, of course, before microscopy, modern microscopy. So he was drawing these microbes that he would see. He called them animalcules at the time. We didn't even have the term microbe yet, and he drew them in these little figures here that he sent to the Royal Society. So it's important to know that microbes have always existed, but on the scale of human history, at least, their discovery was relatively recent. I actually went, just as a quick side note, to the very site where van Leeuwenhoek first saw microbes through those simple microscopes. So this is in a town in the Netherlands called Delft. And there's a building here now where there's an abandoned storefront, but right by this plaque is where his original house stood. I think it burnt down in the 1930s, but this is the actual parcel of land where he first discovered microbes. And so I kind of went on a pilgrimage there and saw that very location, which was a special trip for a microbiologist. Something else about microbes that I feel is particularly important and that will come into play when it comes to explaining the projects with students in my classes is that microbes are individually microscopic, but they often manifest in macroscopic communities. So everything I told you so far was that microbes are too small to be seen by the naked eye, and that's their very definition. But when they amass in large numbers, like by the thousands or by the millions or even by the billions, they become visible to the naked eye. So I see this sort of basic fact as a spectrum that looks something like this. You have your individual microbial cells. Almost all bacteria are individually microscopic. But then there's this kind of spectrum where they get together in large numbers for a variety of reasons in nature. And those are structures that I call collective life cycles and lifestyles that are all visible to the naked eye. These are things like lichen that you can see right here on campus, colonies of lichen. Mushrooms are just a couple of examples of macroscopic manifestations of microbes that we could see right around here. And there's a whole bunch of other specific examples of these structures here. So keep that in mind that even though they're microscopic, there's a lot of cases where you can see them both in nature, and also you can take advantage of this fact in the laboratory when you're growing microbes as colonies. So this next movie both illustrates what a microbial colony is, like I was just describing, and it also illustrates the next point that I want to make about microbes generally, which is that they're everywhere. So this right here is a handprint. I think you can already tell by now because it's played a few times that this is a handprint. It's actually my handprint. So what this is it's just a big Petri plate, a large square Petri plate. And again, for anybody, many people on this call that are not microbiologists, I just want to make sure that everybody knows what these colonies are and how I grow them and what this substance is inside of the plate. So everything you see in the background in this kind of yellowish tinge is what we call agar growth medium. It's just a semi-solid growth medium that you pour into a Petri plate, and you design the media and set up the media so that it has everything that the bacteria need to grow. So it has water, and it has usually a carbon source, a nitrogen source, phosphorus source, etc. But when you take common objects, like for example, your hand, and I just pressed it against the surface of the agar medium, what then happens when you incubate it is that all of the little bacterial cells on my hand that are invisible now have a chance to grow and thrive over a few days. And they multiply into such high numbers and form these larger structures that we call colonies. And so that's the perfect example of when these individually microscopic microbes become visible to the naked eye. And so I'll show you a lot of pictures of colonies and work that my students do with these sorts of colonies. So every one of these little dots here would be made of probably millions of individual bacteria. There's also yeast that we typically find on our skin. So some of these colonies here would also be yeast. So they're everywhere. Specifically, my background, and then going into how it informs my teaching style now. In the Coulter lab, where I showed you in the beginning, before I had to swap out the presentations, I was the lab imaging specialist. So like I said, it's kind of it was a special lab in that there were about a dozen or so postdocs in a similar career position as me. Like I said, all working on those different projects. And what I did is I sort of worked across just about all of the projects in the lab in my role as the lab imaging specialist. So I was working on time-lapse methods, ultra-high resolution, composite imagery, making 3D models of all these sorts of microbial structures and behaviors and phenomenon related to lots of different projects happening in the lab. So that was the research side of my work in the lab. Meanwhile, I was also involved in a lot of microbial science education. So I was working on several museum exhibitions. Also the book that was mentioned in the description of this event, and doing a lot of blogging for the American Society of Microbiology and other professional science blogs. So these two sides sort of my work in the Coulter Lab really worked together synergistically, because a lot of the imagery I was generating through the research projects was also just happened to be very beautiful microbial forms that we could present in the museum and in the book for people who are not microbiologists to learn about microbiology. So in particular, as it comes to my teaching philosophy today, probably my explorations for the book, Life at the Edge of Sight, and also for the museum exhibit called Microbial Life that's actually still open at the Harvard Museum of Natural History, even though the whole museum is closed right now still, but the exhibition has been ongoing now for 3 years. For these projects, I traveled extensively to collect both images and stories for the book and for the museum exhibit. So this is where I really got this sense of microbial exploration, and also other aspects of microbes that I tend to really focus on today with my students. So I'll give you a few examples of that. So I went to Yellowstone National Park, for example. I've been there many times now, actually, but this was one of my trips, and that's my brother Andrew there, who went on the trip with me. And we're at this site here called Grand Prismatic Spring at night, and he's actually looking over with the headlamp. This whole entire region here is all made up of microbes. So this would be all one big visible microbial mat. So when I said microbes become visible to the naked eye, sometimes they actually do this on a huge landscape level scale, and there would be trillions of individual microbes there. So here's another view. Now you can barely see my brother's headlamp in the distance, and you can see some of this natural orange pigment. This is from mostly the cyanobacteria that are making up this microbial mat. And I'm not going to go into these stories because that's not the main purpose of this talk. But the point is, I was traveling to collect these remarkable stories, and I could give a whole talk right now, a whole separate talk on how being around a thermal area or a thermal pool like this one here in the foreground, how I could tell you about the origin of life on Earth based on a place like this, based on these microbes that live here. So if we went in and took a little sample of anywhere on this microbial mat, you would see this community of microbes that looks something like this. So I could tell you a whole story about the origin of life on Earth and how people are working on places like Yellowstone and hot springs to think about how life first arose on Earth. I could also talk about life on other planets and moons. So places like Yellowstone and these exceptional thermal areas are really good proxies for thinking about the conditions on other planets and moons in our solar system and beyond. So we could talk about that. I could also talk about an enzyme that changed biology forever. So the previous two examples were kind of, foundational science stories, but in terms of a practical use of a microbe, in the 70s, people actually discovered a certain enzyme first in Yellowstone Park called polymerase. And this polymerase went on to revolutionize molecular biology and really biology generally, because of the influence that molecular biology has had on the entire field of biology. So it led to us being able to do things like polymerase chain reaction and sequence genes, and really led in many ways to the entire genetic revolution of biology. And the point is that enzyme was first discovered from a microbe that lived in Yellowstone. So that's a whole nother story that I could tell you on this sort of exploration into Yellowstone. Another example of a place that I went was in England, on the coast of England, this called the White Cliffs of Dover that you might have heard of. These are these massive deposits of natural chalk that are found both on the English and on the French side. There's actually a subterranean deposit of natural salt that goes underneath most of Europe. So I traveled here to collect images for the book and for the museum exhibit. And from here, I could tell you a whole story about these ancient microalgae that actually comprise most of the makeup of this natural salt, and the stories of these ancient microalgae. So here's the big cliffs, you know, tons and tons of this natural chalk. This is what the crumbly chalk looks like closer up. And then with a scanning electron microscope, you can see these are actually the fossilized shells of these microalgae that were once living in the ocean millions of years ago. And then through long geological processes, they became kind of entombed in the natural chalk at the White Cliffs of Dover. So we could have a whole story about that. Related to that, this is what some of those microbes look like today in the ocean today. So they actually form these huge blooms of microalgae. So these blooms that you're looking at right here, this kind of bluish green swirling in the ocean, these are sometimes hundreds of kilometers long, and they're made up of similar microalgae as the ones that became deposited in the chalk in Dover. So in terms of useful applications or aspects of microbes, I could certainly talk all about the fact that these marine microbes actually make about half of the oxygen in the atmosphere that we breathe today. So that's certainly a main positive aspect of microbes and these marine algae and other marine microbial organisms in general, the fact that they produce about half of the oxygen in the atmosphere. Coming away from, you know, these satellite images and these kind of big grand places like Yellowstone and the cliffs of Dover to some place that might be a little bit more familiar. I also traveled to regular familiar places like this, which is an artisanal cheese shop in Boston. And we can look at some of the stories that I could tell you based on just based right off of your kitchen counter or right off of the counter of a cheese shop like this. So if you like cheeses, you've probably been to a cheese shop like this and you see all these cheeses and all with their rinds. And these are all microbial products. So the curd is made by microbes. And then here, looking at a cross section of blue cheese specifically, all of the pockets inside of blue cheese are made up of this particular mold called Penicillium roqueforti. Originally, it came from a particular area of France, and that's where it gets its name. So the blue color and the characteristic flavors of blue cheese come from this one Penicillium species. Maybe when I said Penicillium, you already made the connection that the antibiotic penicillin also came from a very closely related species of mold. So this is a filamentous mold type of fungus. So from this piece of cheese in this really regular kind of familiar environment, I could tell you whole stories about these revolutionary antibiotics like penicillin that come from these mold species, or about how this mold contributes to the delicious flavors of blue cheese, for example. Another beautiful and important microbe for food production is called Aspergillus oryzae. And here's some pictures of Aspergillus growing on grains of, on rice grains. So these are actually, just for a sense of scale, this is an individual piece of rice here. This is a fungus that makes sake and soy sauce and miso and a lot of other foods like that. So similar story as the cheese. And I could also, just for fun, tell you stories about the little little animals, actually near microscopic animals that live in some of the cheeses that might be your favorite cheeses, such as cheese mites. So this is a little cheese mite here crawling in a piece of blue cheese. And you can find these in just about any aged cheese. And in our book, we kind of go into this fantasy story about somebody who shrinks down into a piece of cheese and then encounters a cheese mite. So they're really like the charismatic megafauna, I guess you could say, within the cheese microbial ecosystem. Those are the largest animals, but of course they are themselves near microscopic. And then I could talk about a lot of other foods like kombucha that are made by microbes. So this is how you make kombucha. This is actually a big microbial community up here. All of this white material that you're seeing is a biofilm. And the microbes that live inside of this biofilm contribute the unique flavors to kombucha, including producing acetic acid, which gives it the vinegary taste and other flavors. And this is what that kombucha biofilm looks like through a scanning electron microscope. So you can see these are yeast, and then there's little bacteria mixed in. And in case you're curious, these strands here are actually all cellulose fibers that are produced by bacteria in the kombucha biofilm. So those are just a little few stories that I could go into more detail about food. Also, another familiar and nearby location, I could go into stories just from the leaf litter, which seems like a very, familiar and maybe you would think boring location, but there's all sorts of microbial activity happening in the leaf litter. And I could tell you specifically about this microbe here, which is called Physarum polycephalum. This organism is a slime mold that forms these macroscopic networks that go through the layers of the leaf litter. Here's a time-lapse movie I took of one of these slime molds from the leaf litter that I brought into the lab. And I could tell you all about how it moves and searches through its environment, how it maps its environment. Some people actually consider these slime molds intelligent organisms. And there are real research groups out there working on slime molds and using slime molds to help us actually build human networks. So they make very efficient routes between their food sources. And people have used these slime molds to, for example, try to reproduce transit routes, or they can also solve mazes, they can make decisions. So there's a lot of both interesting and practical applications of this humble slime mold that comes from the leaf litter. So those were a few examples and a sense of these exploratory trips that I went on for the book and for the museum exhibits while I was in the Coulter lab. And now it's really all about applying my experience and this approach to my environmental microbiology and bioremediation courses at Paul Smith's. So I should say I also teach exploring biology. I also teach the intro bio labs, and I certainly apply this approach to those courses as well. But most of everything I'll talk about from now on will apply to environmental microbiology and also bioremediation. So specifically coming from those stories I just told you, I have in my teaching approach a focus on the macroscopic side of microbial life and on identifying microbes based on their colony or community level features. So this is a scattering of a bunch of microbial colonies showing some of their natural pigments. And this actually is a practical way of doing microbiology in the labs here, because a lot of microbes can be identified at least at the genus level based on their colony morphology. So it's an easy and very approachable way to work with and to identify different microbes. And I think you could already see why I have this focus on the macroscopic side of microbes based on my introduction. I also continue a focus on microbial communities and multicellular behaviors. So this is something that I find particularly exciting about microbiology, and I definitely use this to try to get my students excited about microbes, because these are the things that people tend to know less about when it comes to microbiology. So everybody sort of envisions microbes as little cells that float around, but not everybody knows that they can form communities and that they prefer to live in communities, and they form structures like this. Here is a biofilm produced by a certain soil bacterium. This whole thing is about the size of a dime, and this is a time lapse showing you how this colony develops. And hopefully, when you're looking at it, you might actually think, is this a tissue? It almost looks like an animal tissue, and actually there's a lot of evidence that these biofilms develop almost akin to animal embryos. There's a lot of basic biology that overlaps there. So I continue that focus. I continue my focusing on imaging and microscopy. So there's an image here of some of my students last year. This was last spring, pre-COVID. So you can see there's no masks here. And so I really try to engage my students, by repeating some of these time-lapse approaches that I've refined in the past, doing a lot of microscopy, a lot of exploration of natural samples that we collect around campus using the microscope. And I also continue a focus on bioprospecting through natural samples for useful applications of microbes. So if you think back to like the fungus that makes blue cheese, things like that. So not just microbes generally, but how can we use microbes from the environment for useful applications that are in human society, and on the positive sides of microbes in general. So probably right now, maybe that's even more important to focus on the positive aspects of microbe. Obviously, there's a microbial entity, microscopic entity right now traveling across the planet that's really causing a catastrophe throughout the global society. So I think probably it's better now than ever to focus on some of the positive sides of microbes as well. So this is just a time lapse from NASA showing these are again the marine microbes in the ocean here, all throughout the ocean, that are producing a lot of the oxygen that we breathe. And I also have a focus on finding microbes in familiar natural places on campus. So one thing that we do in my classes is these prints. So like I showed you the handprint before. So if we take any leaf from around campus, any kind of decaying leaf, and also press that onto an agar medium, you can see kind of this microbial imprint or microbial shadow left behind by all of the microbes that were deposited on the medium, similar to how I described for my handprint. So that's specifically the ways that I focus my past experience on and move that into my teaching approach today. So specifically, the projects that I want to talk about for the next 10 minutes or so, 10, 15 minutes until the talk is over. Number one is in my bioremediation course. We are trying to culture bacteria from campus, soils and waters that can so-called eat oil and other hydrocarbon substances. So first of all, again, I should explain what bioremediation is, because I don't want to take for granted that everybody here knows what bioremediation is. So a very quick definition of bioremediation would be using organisms to clean up environmental pollution. So like I said, most biodiversity, like I said in the beginning, most biodiversity is microbial. So most of the species on the planet are bacteria, fungi, and other microbes. And within all of those microbial genomes out there in nature, these maybe up to 1 trillion different species, is a natural genetic ability to break down just about every chemical that's known to man. So that's a major useful application of microbes that are living right here on campus, is trying to find the microbes that we can use to break down pollution. So bioremediation is one type of remediation activity that's essentially addressing the side effects of human industry, particularly in, from around the 1800s through about the 1970s. There was a long period of time when many companies just dumped their waste right into a river or right into a pit in the ground. And there really just wasn't any widespread understanding that that was a bad thing. And now, of course, we know that a lot of those chemicals are really dangerous to human health and the environment. And so what we're doing now is trying to go back and clean up all those sites where we did all that damage before modern regulations were in place. So I put a couple of examples here from local stories related to environmental pollution. So we have, for example, this cleanup site on Lake Flower that was there a couple of years ago. So there's an article here in the Explorer about that Superfund site there. This Superfund site actually was caused by this old oil gas company that was located here in Saranac Lake. And they were producing a certain type of oil gas that was used for lamps that you would have like outside of your house. And in the process, they produced a lot of chemicals, dangerous chemicals that fall into a group of chemicals called BTEX chemicals. And they actually leached out through Brandy Brook here and ended up being deposited in high concentrations in Pontiac Bay. This is actually directly the site where the big new hotel is. So right after they cleaned up that site, they started developing it into the hotel site. So that's one example of a local story. Another one just came up actually a few days ago. So this headline here comes from October 26th. State says Saranac Lake Airport is contaminated by a group of chemicals called PFAS. So I put that in here because like I said, it's super recent and relevant story. And now the state's going to be, has identified the Saranac Lake Airport as another Superfund site, and there'll be efforts to clean up these PFAS chemicals. These were actually chemicals produced from firefighting foam. So in drills and exercises, you would release a lot of firefighting foam, which all the while had these PFAS chemicals that don't readily break down and also cause health problems. So they're finding it in high concentrations now around the airport and also in the groundwater beneath the mobile home park that's right there. So obviously a big potential influence on that community. So in this project, my students have been culturing bacteria from campus and testing their ability to biodegrade, for example, crude oil. So we could apply the same approach that I'm going to explain here to really any type of chemical, but I'll just say that I really avoid any kind of working with any kind of chemical that's too dangerous in the lab. Unfortunately, a lot of these chemicals would have to be used in a special fume hood, or they could release volatiles, and really wouldn't be something I would want students to work with. But we can work with things like crude oil. So there was actually already some samples of crude oil like this that were already on hand in the lab. So we go around campus and find different soil sites and water sites. We collect that soil. We usually have to dilute it a few times, and then we just directly plate it. So take some of that liquid and put it again on top of one of these Agar Petri plates. And these are a few examples of some of the bacteria that we're currently working with in a project that's actually ongoing right now. So we have one isolate here we call isolate A. This is a particular type of bacteria called Actinobacteria that we already knew we were interested going into the project. So we sort of tried to culture an Actinobacterium because we know that they can break down a lot of chemicals. Isolate B is this one here that has this spiral-like colony morphology. So this is a really good example of where we can identify a microbe based on their visible colony morphology, because this bacillus grows in these multicellular spirals. And then we're also working with this purple bacterium here that has this natural purple pigment, which is likely bacterium in the genus Genithobacterium. and then also this orange pigmented bacterium here. And these two came from lake water. So these two on top came from the soil, and these two came from lake water. The pigments are nice and often just make it easier to see and work with the microbes on the Petri plates. This is another picture of Bacillus mycoides, just a close-up. That was isolate B here, showing you again this really characteristic spiral multicellular behavior. So this is an experiment that I'm going to explain briefly, which actually this image came from the lab today. So I took this image today, and that was why the presentation wasn't updated with these slides from Dropbox. That's why I had to reset it. So this image actually came from lab today. Each row here was completed by a different student. So students are working with these 4 different isolates. And I won't go into too much details of the experiment, but basically what we do is we have a medium here in the background that is devoid of a carbon source. So that should limit the growth of the bacteria. And then we make a little well in the center by punching out a little well. And then in the negative control, we add just water to that plate. So in all the plates over here, we would expect to have just a little bit of growth, maybe left over from internal stores that the bacteria have, but not any kind of robust growth. Here, these are 2 replicates of a positive control where we added glycerol in the center. So this is a carbon source. So now we should see growth. We're hoping to see growth on this positive control. And you can see that here as this really fuzzy layer of bacteria for isolate A. And then the experimental condition is where we add the oil in the well. And actually the whole reason that we set this up with the well is because the oil doesn't dissolve into the medium. Otherwise we could just add it directly to the medium, but because it would just sort of bead up and not make a nice plate, that's why we do it this way by making these wells in the center. So essentially what we're looking for is to see no growth here, growth on the positive control, and then if we see growth also in the plates with the oil, then that's good reason to think that these microbes are actually eating the oil, which means they're using it as a carbon source, which means they're breaking down the crude oil. So in that first row here of isolate A, we have some really promising preliminary results. These are also results that came from my lab earlier today. So this again on the left is the negative control, so no growth here. This is where we added water. This here is with the crude oil in the center, and you can see we got good growth here, which is indicating that this isolate, and if we go back here, remember this was this one from the soil, is looking like it's able to biodegrade the oil. Here's another higher resolution image of that plate. And what really reassures me that we're seeing real growth on oil here from this isolate is the fact that we're noticing this really robust growth that's occurring right around the central well. So this is a close-up around that central well. And in even a further close-up, you can really see how the bacteria, which are all here in white, this would all be made up of bacterial cells, are really just growing right through and all around the oil. So we're following up on these experiments, but I think of those 4 isolates, we've at least have one that's able to biodegrade the oil. Project 2, I'll go through briefly, is related to the first project. So here we're also exploring the soil for different bacteria with a slightly different purpose. Here we're looking for species that produce antibiotics. That also has a nice visible phenotype on a plate like this, because here you can see one species that's likely producing antibiotics, and it's actually created a clearing zone here. It's killing off all of these other bacteria and mold on the plate. So that's a dead giveaway that this colony in the center is actually secreting antibiotics into the medium that are inhibiting the growth of the microbes around it. So these are candidates for species that we want to build up into a whole collection of these actinobacteria that produce antibiotics, because these species from the soil are actually nature's medicine makers. So here's some other close-ups of what these actinobacteria look like. They often have natural pigments as well, so that's what you're seeing here in this purple-blue coloration. And they produce actually about half of the known clinical antibiotics that we use today from the doctor come from this one group of bacteria. And there's a lot of interest and reason for going out and looking for more new types of antibiotics produced by these bacteria, partially because we're seeing resistance to a lot of the antibiotics that we already have. So we kind of have to continuously search and discover new antibiotics in order to fight off pathogenic bacteria. So this is what you're seeing here. This is also, this species here is an Actinobacterium that's inhibiting the growth of this yellow bacterium around it. So again, it's a really visible and easy phenotype that students can see from soil cultivations. This is a time lapse of an Actinobacterium, and you're seeing the colony develop, and you're also seeing some blue pigment that's released into the medium. And in this case, for this species, the blue pigment is also the antibiotic. So something else that makes this, these bacteria easy to work with in the lab is that oftentimes their pigments are colorful, or their antibiotics are pigments that are very colorful, and you can easily recognize them on a Petri plate. And these are just some of the structures of the various antibiotics that people have already discovered from these bacteria. The bacteria are really fuzzy, like I've been showing you, and there would also be antibiotics in all of these little droplets that form on top of the bacteria. So yeah, many people might not know that about half of all clinical antibiotics come from these soil bacteria, and the idea is to generate a whole collection of these bacteria from local soils, because of course the Adirondacks are very, very rich in diverse types of soil. Project number 3 is to make sustainable biomaterials from fungi and agricultural waste materials. So here's a picture of some of my students going out and canvassing through the forest, looking for different fungi. The idea here is related to bioremediation, because this sort of project is aimed at eliminating styrofoam and other types of packaging materials from various industries. So these materials that we're trying to make using fungi could actually replace a lot of packaging materials and then start to eat into those really harmful styrofoam industries and other types of plastic packaging. So in step 1, we just go out in the woods and we collect a small tissue sample from various types of fungi. So these are all fungi that we found this fall. We then take a little, so this is a coral fungus here. We take a little piece of the mushroom tissue, put it on a certain medium that's meant for culturing these fungi, and then this is called cloning. So we're cloning the wild mushroom and getting it to grow in a culture in the laboratory. So that would be the next step here. So we've been successful at culturing many of these different mushrooms. And then in step 3, we make a mixture where we mix the fungi with a substrate, which is usually a waste product like agricultural hemp waste product or sawdust or even coffee grounds. You just add some water and a little bit of sugar with the waste material and spike in the fungus, and then you pack it into a growth form. So these would all be called growth forms, and you can make all sorts of different shapes and materials out of this. Once it grows in these growth forms for about a week, you then actually just bake it in a normal oven, and that cures the form, and then you end up with a pretty permanent final product here. So this is actually a flower pot that one of my students made, and this is more of just like a brick material. And so this material is made of nothing other than the fungus, which makes this network of mycelia that makes it really strong, and then an agricultural waste product. So we're doing experiments with different fungi and different substrates and trying to make these products. Here's an example of where we're going forward with this. This is my last slide. The idea is that we can have a system going where we use both local fungi that we cultivate from around campus and also sawdust that's a waste product of the Paul Smith's mill. So this was one experiment where we used maple sawdust and then a turkey tail fungus that we had cultured and left it to grow for a while. And you can see it did make, at least in this beaker, this sort of composite material that we could refine into and pack into a different growth form to make a certain material. This is also relevant to jobs for students because there's actually a company in New York called Ecovative outside of Albany that now has big contracts with 3M and IKEA, and they're really, they've actually taken off, and they have now hundreds of employees for specifically making these types of fungus materials. So this is not something that's just kind of a curiosity or something that we're doing in our classes. This actually represents an emerging industry that, like I said, we hope will start to replace styrofoam and other more harmful materials. And with that, I will thank you and happy to answer any questions that came up through the chat or otherwise. And again, I'm sorry I had to swap out that presentation in the beginning. But thanks again, and let's see if there's any questions. Where's the chat? All right, so I see some comments here. So yeah, one other thing I'll say, if anybody's waiting to ask a question, is that people have actually made, for example, a canoe out of these mushroom materials. So that's probably the most extreme, one of the most extreme products of these mushroom materials. A student at another university made a full-size canoe out of these products and then took it out into a lake and tested it out, and it held water. These materials are actually pretty waterproof, and they the neat thing is they last in the environment unless you break them apart. So right now, if you just sort of threw this into a compost pile, it wouldn't break down. It would be pretty tough. But as soon as you pulverize it into small pieces, then different bacteria and other microbes can gain access to those small pieces, and then it breaks down within about 30 days. So it's both compostable, and it also uses up agricultural waste products. So we've been thinking of some different things that we could make that would be relevant to Paul Smith's. Like, student had a really good idea to make bobbers, for example, from this mushroom material, which seems like it would really address, a problem of eliminating plastic bobbers, that if you lose them in the lake, they're there, forever. And also something that's very doable, because we could make a basic mold for a bobber, just add some little hook in there, and something that we could actually feasibly produce in high numbers. So that's a couple of ideas that students have had for what to make. We've also considered making like a statue or something more artistic on campus as a final product of this mushroom material. So let me know if anybody else has any ideas of things we can try to make. People are actually trying to make entire villages out of these mushroom materials, if you can believe it. People have made buildings, and some people even think that our cities will be made out of mushrooms someday. So I guess the sky is really the limit. So I'm not hearing any questions. I'm going to check the chat.
Scott, there's this question here from Doug Brush, I'm sorry. Would there be a concern with causing an invasive species situation if you use fungi to make packaging and it ended up in another country or continent? Could it cause a problem?
Yeah, that's a good question, and it is a concern that comes up with sometimes bioremediation projects in general. People, concerns about releasing microbes into the environment. Specifically with these sorts of products, this is actually made from an oyster mushroom, from an edible oyster mushroom strain. So all of the mushrooms that we work with, or fungi that we work with in the class, are you know, non-toxic, non-poisonous species. There's plenty of those where we wouldn't have to experiment with dangerous ones. So first of all, they're safe to begin with. But the real important point is that once you bake it, you actually cure and kill the organism. So you're only left with a hardened version of the mycelial network, it's called, of the fungus, and it's no longer living. So it actually would not produce any new mushrooms or any new kind of biomass in whatever environment you released it into. So that's one of the benefits, is that it becomes actually inert through the curing process. But that's a good question.
We also have a question here from Kate Glenn, wondering if you're going to be publishing the results from some of these research projects.
Absolutely, that's the plan. I would say that right now my students and I are not trying to be too ambitious in these first couple of semesters. So one thing I didn't mention in the beginning is that I started here last August 2019. So I've only been here a little bit more than a year. And so really what I'm trying to do with my courses so far is get these projects up and running. But certainly I do hope that the combined efforts of many students over, you know, several generations of classes will lead to tangible published papers and things like that. Right now we haven't started publishing. We're just kind of working on proof of concepts, I guess I would say, for these 3 different projects. But absolutely, that's where we would like to go forward in the future. I agree with Jory that it would be great for students to share the results at the research, ADK Research Consortium. So we will definitely plan to do that, particularly if we continue getting a good result from that bacterium that looks like it's eating the oil. So that seems to be a really promising result, and that's something we would definitely want to continue following up on. You know, sequence some genes to actually identify the bacterium at the species level, and then figure out what it is. And we could even look into its genome and see what genes we think might be responsible for breaking down the oil. And I agree that would make a great presentation. So I don't know what you think, Heather, but if there's no further questions, we could end the talk, and I could just say that I'm happy to answer any further questions that come up through email. You could reach out to me anytime. And yeah, look forward to seeing some presentations from students in the future from some of the products of these projects.
Okay, well, thank you, Dr. Chimileski, for joining us this evening and sharing some pretty interesting stuff. I think I'm going to have to go check out that book. In case you didn't know, Dr. Chimileski's book is available at the Paul Smith bookstore, the Pack Basket, and you can purchase it online, or if you happen to be lucky enough to be on campus, you can stop by the bookstore. We do have other Smitty Story Hours coming up in the near future. November 12th, we'll have a conversation with Curtis Hem, class of 90, and Paul Sorgel, class of 1970, 2 culinary arts former faculty members, and of course, alumni. And we'll have additional programs coming up in December, January, February. We're trying for once a month, but we're getting a lot of topics, so we might wind up with adding 2 a month. We'll see. Thanks again, everyone, for joining us, and we'll talk real soon. Thank you.
Thank you, Heather, for the invitation.
Thank you very much.
Bye-bye.